Garriga, and today I want to explain my PhD project, the regressive alignment. It's a new way to perform a multi-sequence alignment. To start, a quick look to the Wikipedia definition of MSA. What is a multi-sequence alignment? It's the alignment of three or more sequences that can be DNA, RNA, or proteins, and we assume that they have an evolutionary relationship between them. Then it's the MSA use in the community. Yes, like if you are working on phylogeny mapping or classification, for example, you are running an MSA on your analysis. But if you don't trust me, if we check the top 100 cited paper on Nature, we can see that number 10 is Clustal Omega, like one of the oldest aligners, and it has like more than 40,000 citations. How MSA works nowadays, like the most common method used is the progressive method, and how it works, like it starts from a guide tree, like a tree like this, that it's clustering the sequence by similarity. These numbers are the length of the sequence; they are not important now. And how it works, it starts from the bottom, start aligning 2 and 1, then we put it here, then we align this 2 and 1 and 6 and 4, it's a profile, then we place it here until we reach the root. Like once you get the root, we have this final MSA. And another point, it's data, how data increase in this era, no? Like 6 months ago, I was talking that we were on the big data era, but nowadays I'm not sure in which era we are. In pandemic era, or I don't know how we call it, but on December 18, when we submit the first draft on bioRxiv, the most kind of highlighted project was this vertebrate genome project, and the idea of this project is to sequence 10,000 species. But when the article was accepted on October 19, we already were talking about the Earth Biogenome project, and we go up to 1.5 million eukaryotic species. Like in less than 1 year, how fast the community grows the data, the necessity. Does the progressive scale as much as the data? No. As we can see here on the Clustal Omega paper, in this figure, people thought that when we have more data, our result will be more accurate. Like more data, more information will be better for our results. But they saw that around in 1,000 sequence, after that the accuracy start dropping. Like you increase data, accuracy increase, but when you reach this point, accuracy start decreasing when you add more data. Why we drop accuracy when we add more data? First, because aligners are based on profile alignment, as we saw before. Like just after first step, everything was a profile alignment, and large profiles contain mostly gaps. Like once you insert a gap on a profile, it's impossible to remove, and like it expands quite fast. Like when every iteration, every step that you do, this gap will go up to 100 or whatever. It's not very friendly with the gaps. And in large column, mutual information is lost. Like when we have a huge amount of column, we miss information from one side to the other side. Then we need to find how we can fix it, how we can try to solve and address these problems. And we have these three strategies. To avoid the gaps, we are trying to avoid aligning the large data set, like we are avoiding to go farther than 1,000 sequence. How we do that? We just split the big MSA in a smaller one, and it's kind of the same strategy for the flattening. Like instead of going with profile, we make a divide and conquer strategy. Like from 1 million sequence, we split this problem in problems of 1,000 sequences. And for the ties, we avoid early decisions, and instead of start aligning the most similar sequence, like we don't know, like we have a picture very restricted with two sequence, we start that they are on the bottom of the guide tree. We start aligning the most distantly related sequence. We start from the root, like kind of we have a bigger picture of what we have below us. And that's why we propose this regressive algorithm. That how it works is quite easy. We have kind of a recipe with 4 steps. Let's start with labeling the intermediate nodes with a representative sequence. We start from the bottom to the top. We have here the same guide tree, and now we will use these numbers, okay? This is the length of the sequence. This sequence length is 2, this sequence is length 1. Now we will label this node with the representative. For us, the representative is the longest one, because we thought that the longest one has more information, more biological information. Then we just compare the length, like 6 and 11, which is longest, 11, that's an easy operation, until we have all the nodes with information. For example, if we go in this node, we can say that below this node, the longest sequence will be 11, and kind of the more biological information will be contained in this sequence. Next step is to collect this representative sequence, and we start from the root to the bottom. As the size of n will be, we use on the article n equal 1000, because we saw the cluster omega, like we can improve accuracy until 1000 and then we decrease. But for here in the example, we use n equal 3. Like we start in the root and we want to collect 3 nodes, 3 sequence. Then we have 14, we expand the child. We have 11 and 14, it's not a node. Then we go in the 11 and we expand 6, 11 and 14. Okay, we have the first subgroup of these 3. Now we go on the 6 and we do the same procedure. We expand 2 and 6, it's not a node. Then we expand 2, we have 2, 1 and 6. Okay, we do the same with the same kind of hierarchy, and we have these green ones. And finally we go to this 6 and we expand 6 and 4. We are in the leaf, like there is nothing below. We just keep these 2. Like it's not a problem that we are lower than 3, because kind of we didn't go through the border. And we have all these subgroups. Like this could be the parent one, the green will be the first generation, and the blue the second generation. Now we are doing our alignment step. Like we align all the sub MSAs with whatever aligner we want. Like we have the guide tree. These are the groups, like the subgroups that we have before. And here in red is the common sequence. Like we can see that between parent and first generation, this 6 is common, this 11 is common, and this 14 is common. And for example, this 11 is common from this and this. Okay, we will use in the next step. But now we have all these subgroups. We call any aligner that we want. We run all these subgroups in parallel on the same time. Like we don't need to wait for one to finish the other. We run everything together, and at the end, magic happens, and we have as many sub MSAs as subgroups we have. Like we have kind of a portion of the result here. And finally, we merge everything. Like we merge all the sub MSAs using the representative. Why? Now we will explain the representative. Like this is the common sequence between the parent and the first generation, and things that is common in both sides, in both generations, we can expand the gaps. For example, P and Q, we have here 2 gaps, but we don't have a gap here. Okay, we expand on the purple side, we insert 2 gaps. It's the same on the other way around. If we have 2 gaps on the purple and not on the blue, we just introduce it here. And if we have 2 gaps on the purple and in the blue, we don't know if it's the same evolutionary event or not. That's why we cannot combine. We have to take everything together. Like we have 2 from the blue and 2 from the purple. And that's all. That's how it works. And it makes fancy and nice alignments. Let's see if it works or not. I think that you need to believe me with more than my explanation. And this is the results. Like that's a plot. On the x-axis we have the time, the CPU time, and on the y the accuracy, total column score. It's not that's the average on HomeFam, like the biggest top 20 HomeFam datasets. They have over 10,000 sequence. And this is the non-regressive, like the progressive flavor. Like we can see all the dots, and what we want is to go up to increase the accuracy, and if we go left, it means that we are faster. And these are the results. For example, with cluster of R tree, we go from here to here, it means that we are faster and we are more accurate. We increase the accuracy with FFTNS1 part 3, the same. We go with better accuracy and faster. But here, for example, we go faster, but we decrease the accuracy. And we have here in the corner these two dots that they are only on orange because it's not able to run GINS1 on progressive. Sorry, like GINS1 it's not able to go to this amount of sequence. That's why we can, like, they are very accurate. It's quite time consuming, but we are able to run very accurate aligners on big data sets. And another point is when it starts failing, like how many sequences it can align until the first failure. With the non-regressive, we were able to go up to 100,000, and in the regressive, we were able to go up to 1.5 millions. Like, but that's the maximum. No, sorry, that's the maximum. We don't know. Like, we were not able to find a family bigger than 1.5 million sequences. It means that we don't know where is the failure of the regressive mode. And it's a good, I think that it's a good point because we thought that our biogenome project was trying to sequence 1.5 million species, and we are going here with 1.5 million sequences. I think that we are, like, the scalability is quite interesting. As a conclusion of the project, of the method, we can say that we can split cluster and alignment fields. Like, we can have a group just focus on clusters and another group just focus on alignment. And at the end, we can use whatever clustering method with whatever alignment procedure we want. Like, the bottleneck of the regressive is the guide tree. Like, if the clustering method can produce the guide tree, we can run the regressive. It means that, and it's quite promising because we can use a clustering method non-biological. Like, we can use some big data or machine learning or whatever clustering method that is in whatever field, and we can try to fit in this aligner. We are almost linear in time, we are more accurate, and we are able, like, more versatile because we are able to use very accurate methods that they are not able to scale. And with this method of divide and conquer, we can use GINS1, for example, on biggest data sets. Take home message, but everyone is at home, but I don't know, keep it for you. The regressive, it's just 4 steps. Label the intermediate nodes, like from the bottom to the top. We collect the subgroups, like from the root to the bottom. Then we align only in this step. On the third step, we align in parallel all the subgroups with the aligner that we want. And finally, we merge all the sub-MSAs without doing any aligner procedure. If you're interested, we already have the publication. You can check it, and yeah, you can run it with Nextflow, Docker, and there is a GitHub repository with all the pipeline to, if you want to plug just your data set or your sequence and see how it works. And that's all. I want to thank Cedric Notre Dame as a PI and Nepal Flowden as a co-author, and all of the members of the group to support and help me with this project. Thanks for all, and yeah, you can try to find more information in the website of the coffee. Thanks.